Take it away.
Hi, everyone. I'm Pedro. I'm a PhD student at Schlinger Lab, and today I'm going to show you how you can use DNA language models to study nucleotide dependencies in genomes and eventually arrive at maps like you see here. DNA language models are trained only on genome sequences to reconstruct a nucleotide given a specific context by assigning a probability to each one of A, C, G, or T for a specific position. And we and others have shown that the reconstruction of the language model highlights functional elements in the genome. But we go one step further and we ask, given a query nucleotide, how can it maximally affect the reconstruction of a target nucleotide? And we derive a value which we term nucleotide dependency, which we then compute for each pair of query target nucleotides in a sequence. And then we plot it in the form of a map where each row is a query nucleotide and each column is a target nucleotide. And we find different patterns across different several genomes. And so here an example of a pattern that we find. For example, you see here a promoter region in S. cerevisiae, and you see the reconstruction on top, which highlights transcription factor binding motifs, and you also see a region poly-D-ADT, which is highlighted by the reconstruction. And if you look at the map, you see that the areas that belong to the transcription factor motifs appear as blocks, which contrasts with the poly-D-ADT. And that is because in a transcription factor binding motif, each nucleotide depends on each other because they are bound by a common factor. So we then derive a score, which is the block score, which classifies how blocky each position is along the main diagonal, and we use it to predict whether a nucleotide is bound by a transcription factor or not. And we check that the performance without any fine-tuning is as good as the experimentally curated PWM. We also find distal patterns like anti-parallel diagonals, which we check belong to non-RNA structure and can very accurately predict without any fine-tuning RNA structure contacts, and we also validate novel structures. We use these dependency maps to benchmark across several, more than a dozen language models. You see here an example for dependencies within tRNA sequence, and while some models are able to capture the contacts of the structure, other models struggle and produce a series of artifacts which have to do with modeling design and also data selection. In particular, models that are only trained on human struggle to find these very conserved but more infrequent elements. We also set out to use nucleotide dependencies to classify variant effects. And on a promoter mutagenesis screen in human, you can see here an example in a human LDL receptor promoter. You see on top the nucleotide reconstruction and below the mutagenesis screen result, and you can see that they correlate pretty well. However, this correlation is not better than, for example, taking alignment-based conservation scores. Then if you use a metric which is based on the average dependency on that nucleotide, we outperform its own reconstruction and we outperform conservation-based scores. So how influential a nucleotide is a better predictor of function than its own reconstructability. If you want to know more, please, I'll be happy to receive you at poster A341. Thank you very much.